my screen for you there. Okay, so thinking back to the other day, we were talking about the two divisions of the autonomic nervous system, providing us with motor output signaling to things like cardiac muscle, smooth muscle, and glands, and which we'd already talked before back at the beginning of the units. We learned that the two divisions, there's a third, enteric, but the two majors that we talk about at this point in the semester are the sympathetic and the parasympathetic. Sympathetic, which we learned produces a fight or flight response, and does so to do things to make us physically active, whether that's as drastic as running from a bear or fighting a bear, or just going for a jog, which people like to do. The opposite division of that is the parasympathetic division, which produces a rest and digest response. Okay, what you guys will be exhibiting right now in the case of resting conditions. So resting heart rate, might be digesting the food that you just had for lunch, and so on and so forth. Okay, we talked about there are two other terms to describe those two divisions. The sympathetic nervous system, we learned, also goes by the term thoracolumbar division, and that's because it uses the thoracic and then part of the lumbar region of the spinal cord. Signals come out typically, and synapsing occurs right in this trunk or chain of ganglia right outside the spinal column. And then the second neuron, the postganglionic neuron, carries the signal the rest of the way to things like the lungs and the heart that you can see there in the picture. On the left side of the page, you can see the parasympathetic nervous system, what we can also describe as the craniosacral division, utilizes 4 cranial nerves. So there's where the cranio, and then the sacral region of the spinal cord. So that's where that term comes from. And as we can see, there is no chain associated with the parasympathetic nervous system. So I kind of posed the question before we left class the other day. How does, for example, your heart know when to speed up when the sympathetic nervous system is active, and how does the heart know when to slow down when the parasympathetic nervous system is active? Well, I think I pointed this out. I don't know if I really did or not, but if I didn't, it's all because of what we see here versus down here. The release of different neurotransmitters is going to be the big regulator and difference between the two. And so that's what we'll start with here today. As noted previously, the sympathetic and parasympathetic nervous systems have dual innervation, meaning they innervate many of the same target organs. So not all organs, but things like your heart and your stomach, they typically are going to have signals coming thanks to nerves for the sympathetic nervous system. There's also nerves carrying signals for the parasympathetic nervous system. Okay, so how do these two sympathetic, parasympathetic nerves produce contrasting effects on the organ? Kind of two things. Okay, number one, the different neurotransmitters that are secreted at the target organ. Okay, so in other words, what we just saw, norepinephrine for the sympathetic nervous system, acetylcholine for the parasympathetic, and then number two, the target organ has different receptors for those given neurotransmitters. So in other words, the target organ has to be able to receive and accept those neurotransmitters. So it's got to have receptors specific for it. So we're going to talk here for the next couple of slides about neurotransmitters and receptors, starting with the one I just mentioned and one that we introduced last week, acetylcholine. Acetylcholine, capital A, capital C, lowercase h, released by what we call cholinergic fibers or cholinergic neurons. So pretty hopefully easy to remember, acetylcholine released by cholinergic fibers. Those cholinergic neurons or fibers include all sympathetic and parasympathetic preganglionic neurons. So remember, preganglionic would be the first neurons, whereas postganglionic neurons are the second neurons. Okay, going back to this illustration. Okay, looking at the bottom 3 illustrations, we see preganglionic, preganglionic, preganglionic. It doesn't matter if it's sympathetic for these two pictures or parasympathetic, it's always going to be acetylcholine, acetylcholine, acetylcholine. What we also see, and this is something we'll kind of talk more about later in the semester, when we talk about scudder muscles, is that acetylcholine is released by the somatic motor neurons that trigger our muscles to contract. All right. So acetylcholine, 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 acetylcholine. It's also going to be released by sympathetic postganglionic neurons that innervate most sweat glands, okay, which is unique because typically when it's the second neuron, postganglionic neuron of the sympathetic nervous system, it's not acetylcholine. The only place where we see the sympathetic postganglionic neurons secreting this acetylcholine is at the sweat glands. I don't really know why sweat glands are why, where we see acetylcholine released by the sympathetic postganglionic neurons, but that's what it is. Okay. And for the parasympathetic nervous system, not only is it the preganglionic neurons secreting acetylcholine, but it's also the postganglionic neurons. So going back once again to this illustration down at the bottom, acetylcholine is released here and it's released here. Okay, so it's twice in a row. Okay, whereas here for the sympathetic, it's acetylcholine and then NE for norepinephrine. Okay. Now, the receptors that receive and accept acetylcholine are called cholinergic receptors. They're integral proteins in the postsynaptic plasma membrane of target cells or target organs. And this includes muscarinic and nicotinic. Those are two kind of types. Muscarinic, that word kind of sounds like muscle at the beginning. And that's where you find it on cardiac smooth muscle, on smooth muscle, and then not a muscle, but obviously what we call glands. And then nicotinic kind of sounds like the word nicotine. Okay, but these receptors are found on the plasma membrane of dendrites and cell bodies of both sympathetic and parasympathetic postganglionic neurons, the chromaffin cells of the adrenal medulla, and at the neuromuscular junction. Okay, I'm going to jump ahead a couple slides, and we can see those different receptors shown here with three very similar illustrations to what we saw the other day. At the top, we've got parasympathetic nervous system, and then at the bottom, we've got sympathetic nervous system and sympathetic nervous system specifically for sweat glands. Okay, looking here, we see once again acetylcholine released by the first neuron, no matter what, if it's sympathetic or parasympathetic. If it's parasympathetic, it's acetylcholine and acetylcholine. Okay, the receptors that are on the postganglionic neurons, no matter what, if we're looking para or sympathetic, those are what we just learned are called nicotinic receptors. Okay, whereas the ones that are on the effector cells, like this, which looks like a smooth muscle cell, could be a cardiac muscle cell, or maybe even a gland cell, it would be a muscarinic receptor. Muscarinic receptors would also be on the cells of sweat glands, which, as you just learned, acetylcholine is released by the sympathetic postganglionic neurons that innervate sweat glands. Okay, this is a kind of rare instance where the sympathetic nervous system releases acetylcholine and again acetylcholine, because typically the sympathetic nervous system does what's shown here in the middle picture. Acetylcholine is first released, and then the second neuron releases norepinephrine. Now, at the bottom there, it looks like the letters A and B, but that's actually alpha and beta. Alpha, you can see it's got this little bit of a tail up here. Kind of always looks like a fish to me. And then beta has a little extra tail. Those are Greek symbols, Greek letters, I guess. Okay, let's first talk about norepinephrine, which was just shown on that next slide. Secreted by nearly all sympathetic postganglionic neurons. The only exception, once again, are those postganglionic neurons innervating sweat glands. But anywhere else, whether this is like at the heart, whether this is at the stomach, okay, if it's the sympathetic nervous system, that second neuron is going to be releasing norepinephrine. We learned the other day that norepinephrine is not only released by neurons, but it's also released from the adrenal medulla whenever we have that fight or flight response kicked in from what we call chromaffin cells. Okay, but along with norepinephrine, our chromaffin cells of the adrenal medulla are also going to release epinephrine. Okay, what we commonly call adrenaline, and if you wanted to, you could call norepinephrine noradrenaline. Okay, in that case, those chemicals are getting dumped into the bloodstream. So we'd say that here they'd be released as a hormone, okay, because they're being dumped into the bloodstream. It's still the same chemical here as far as norepinephrine goes as it is here. Okay, it's just who's releasing it and where it's going. That makes it either a neurotransmitter like it is here or a hormone like it is here. Okay. Now, any neuron that releases norepinephrine is called an adrenergic fiber or adrenergic neuron. So that would be like that one that I just showed you. Okay. As I just mentioned, norepinephrine can be either released as a neurotransmitter by those sympathetic postganglionic neurons, or it's released as a hormone from the adrenal medulla. Whereas when we talk about epinephrine, we're describing it being released as a hormone because it's getting dumped in the blood from that adrenal medulla. Both norepinephrine and epinephrine bind to receptors called adrenergic receptors. So it's adrenergic fibers that release norepinephrine, adrenergic receptors that receive it. Okay, and same thing for epinephrine. We find them on visceral effectors innervated by most sympathetic postganglionic axons. And so that would be what we see here for this effector cell on the next page. Okay, now there's two major types of adrenergic receptors, alphas and betas. Okay, and there's actually subcategories of these. But typically, alpha adrenergic receptors have excitatory effects, like an example here where blood vessels constrict when epi and norepi bind to it. Blood vessel constriction is where blood vessels are like, when you kink a garden hose, it pinches it shut. And that's going to happen when the muscle in the wall of the blood vessels, that smooth muscle contracts. So it's a contraction that makes it excitatory. Okay. Beta adrenergic receptors can have different effects depending upon where you're at in the body. For example, the first one here, dilating smooth muscle in the pulmonary system, meaning we're going to increase airflow into and out of the lungs, involving what we call beta 2 receptors. This is an inhibitory response. Okay, and I say that because the smooth muscle is relaxing. So the muscle's not contracting, it's relaxing. That's an inhibitory response. And that's great. We don't think inhibitory is a bad thing. Okay, by opening up your airways more, you can get more air in and out of your lungs. It's a good thing. Okay, especially when we are, fighting and flighting. Okay, down at the bottom, stronger and faster heartbeat shows an example of a excitatory effect, okay, or an excitatory response. In the case of the beta 1 receptors that are in our heart, okay, when epi and norepi bind to that set of receptors, the heart beats more forcefully and at a rapid rate. Both of those are excitatory responses. So does that make sense how and why I said they can have different effects in different parts of the body? Okay, good. Now, as far as this set of illustrations here on the next page, I like it because it really, gives you a good view of the different receptors, the different neurotransmitters. As you guys know, those of you who are taking anatomy, I love pictures in anatomy class. I don't necessarily do that nearly as much in this class, but if I go back to this slide, which I've referenced several times, not only today, but the other day, okay, this is a rare instance where I'm telling you right now, there's a good chance you're going to see one or two of these images next week. Okay, so what that means is you should be able to just recognize what division of the nervous system each illustration is showing you. Okay, at the top, this is one neuron going all the way to skeletal muscle, so that has to be the somatic nervous system. Okay, the second picture is showing you the sympathetic nervous system, where you have a short preganglionic and then a long postganglionic. The third picture, is another picture showing you the sympathetic nervous system, and you can see what we call the adrenal medulla in the middle. If you show the adrenal medulla, you have to be dealing with the sympathetic nervous system. And then down at the bottom, it's a long pre and a short post. So the first neuron is really long, the second is really short. That's going to tell you that this is parasympathetic. Okay. Aside from recognizing which division of the nervous system's illustrated, you might also have to know which neurotransmitter is being released where. Okay. So, if I were to put this image on the exam and say, which neurotransmitter is being released here, or if I was looking right here, I'd say which neurotransmitter is being released here. You should be able to recognize that. The good thing is, there's a good chance acetylcholine would be your answer. Okay? It might not be the correct answer, but if you don't know the answer, if you guess acetylcholine, it might not be a bad, guess, just because look at the page. Acetylcholine here, acetylcholine here, acetylcholine, acetylcholine, acetylcholine. Something like 80% of what you see here on this page is the release of acetylcholine. The only places it's not is here and here. Make sense? Okay. So hopefully you just can recognize, okay, that's sympathetic. That means it's not acetylcholine here. But if you can't, you know, you can guess, you might get it right. Now, last week I mentioned that certain medicines and chemicals can interact with neurotransmitters and the receptors that receive those neurotransmitters. And a large variety of drugs and natural products can either selectively activate or block cholinergic or adrenergic receptors. We can throw them into two categories, those that are an agonist and those that are an antagonist. An agonist is a substance that binds to and then activates a receptor, which basically means that it's going to mimic the effect of the natural neurotransmitter or the hormone. So in other words, basically, it's a chemical that instead of being acetylcholine, maybe it binds to this receptor and triggers that cell to do whatever that acetylcholine would do naturally. Okay, so it acts like the neurotransmitter, it just actually isn't. Okay, an example of this is phenylephrine, which is found in and causes or helps with nasal decongestion. You guys ever heard of Mucinex before? Phenylephrine is one of the leading chemicals in Mucinex. Okay, if you are suffering from sinus pressure and a cold and you're, you know, like draining mucus all the time, you might take Mucinex or something like that. You might find on the box phenylephrine listed there. What it does, it constricts blood vessels in the nasal mucosa. So the blood vessels that line the inside of your nose become blocked and thus reduces the production of mucus and relieves your nasal congestion. And the opposite of an agonist is what an antagonist does, a substance that blocks or binds to the receptor and then prevents the natural neurotransmitter or hormone from exerting its effect. So essentially, it occupies the space so that like norepinephrine can't bind to the actual cell and thus produce a response. Okay, you guys have ever heard of beta blockers before? Okay, beta blockers are very commonly prescribed medicine. A lot of people are on them because there are a lot, unfortunately, of people with hypertension or high blood pressure. People that are at high risk for things like heart attack and stroke, a lot of times will be on beta blockers. It blocks those beta 1 receptors that I was just talking about in the heart. And that's going to prevent the action of epi and norepi, which, as we just learned, epi-norepi binding to beta 1 receptors increases heart rate and force of contraction. So if you can't bind epi-norepi to those beta 1 receptors, it reduces heart rate and force of contraction, and both of those are going to result in a subsequent decrease in blood pressure. So if you've ever heard beta blockers before, now you know what they actually do. Okay, next couple slides, I just want to talk specifics on what happens to specific parts of the body with the sympathetic and then the parasympathetic nervous systems are active. A lot of what you see here on this slide and the next slide should be pretty common sense, because I've been hammering you on fight or flight, rest and digest for the last week and a half. Once again, the sympathetic nervous system produces that fight or flight response. We don't just kick it in for nothing. It has to be elicited because of stress. So stress increases the sympathetic nervous system or enhances it, which enhances the fight or flight response. In order to fight off stress, whether that's as crazy as running from a bear or just, the stress of exercising, we need energy. And as we know, ATP is our body's energy. So we need to ramp up our production of ATP. Okay. Other things that we do, we dilate our pupils. We make it better to see. That helps us fight that bear off or run for, the jog that we're doing. We increase our heart rate and our blood pressure, so our blood flow is going to increase throughout the body. We dilate our airways so that we are breathing more oxygen in and getting more CO2 out. While we're doing these things, we're going to shut down blood flow to our kidneys and gastrointestinal tract. So in other words, we're not going to send blood to our kidneys to make urine. We're not going to help participate in digestion, because if you think of it like in a drastic situation, like encountering a bear, is it important to make pee? Is it important to digest your food if you are in that life and death situation? Obviously, the answer to that is no. So that allows us to then devote all of our most important resources, they're the things that are going to help us out. Okay, the things that work when we are in that fight or flight response are our skeletal muscles, our cardiac muscles. So we increase the blood supply to them. We're also going to be sending more blood to our liver and our adipose or fat tissue. And that's because in our liver, we store what we call glycogen. Okay, glycogen, we learned in our last unit, is the storage form of glucose. And if you break down your glycogen, that's what this process is called. We're going to talk more about that later on the semester. That increases your blood glucose levels. And really, I should put this phrase right there at the end of this sentence too. So if you wanted to write that out, it might not be a bad idea. By increasing blood glucose, that makes it available for ATP production. Okay. Adipose tissue is just a fancy term for your fat tissue. Okay. We increase the blood supply to that because what we're going to be doing at this point is breaking down our fat. That's what lipolysis is. And that allows us to use that fat for ATP production purposes as well. Okay, so those are kind of just short descriptions of what happens when the sympathetic nervous system is active. Let's now talk parasympathetic. Kind of 5 major things, if you can remember this, what is it, acronym SLUD. That will help you remember those 5 things. Some of those words probably are pretty easy to figure out, or you've already known them for a long time. One of them, I bet, might trip you up. I'll explain what each of them means in case you don't know. Okay, salivation would be the production of what? Saliva. Okay. Urination would be the production and release of urine. Digestion would be the breakdown of food. Defecation would be the release of that food that we have not absorbed. In other words, we're pooping. Anybody have any idea what lacrimation is?
Mother lactating?
That's a good guess, but no. That would be called lactation, actually.
Oh yeah, but I didn't know.
Your lacrimal glands are your tear glands. Okay. Just above our eyes are where you find your lacrimal glands. Lacrimation is the production of tears. Now, you guys are all demonstrating the rest and digest response. I don't see anybody sweating profusely, breathing rapidly, or anything like that. If I look around the room, I don't see anybody crying today. Hopefully that's the case again on Tuesday during the test. Does that mean you're not producing tears?
No.
No, you absolutely are in a rest and digest response. You're just not producing a ton. I know nobody likes this word, but we produce just enough tears to keep our eyes moist. Okay, just that word everybody hates to hear. Okay, you guys all probably experienced times where your eyes get dry and how uncomfortable that is. Okay, your lacrimal glands keep your eyes nice and wet. Okay, and then if we experience an emotional response and we produce a lot more, then we start crying. But that's something completely different than what I'm talking about right now. Okay. In other words, when our parasympathetic nervous system is active, we're conserving and restoring body energy. Our digestive and urinary functions are elevated, and our body functions supporting physical activity are depressed. Okay. So those are just kind of 2 slides describing kind of just general things that are happening. For some very specific descriptions, table 14 here is 15.4, excuse me, is on the next 2 slides. It's actually kind of a 3-part table. I've crammed the other 2 parts here on the following slide. It basically goes organ by organ and tells you specifically what the sympathetic nervous system does to it, what the parasympathetic nervous system does to it. But these 2 tables, I mean, it is very intense, a lot of information. I'm not going to directly drive anything from this and put it on the exam. Okay. A kind of simpler version of that is this illustration on the following page, which I will definitely ask you to know and may pull stuff from it on the exam. More likely I will pull stuff from this and put it on the exam. So make sure you can understand. On the left, it basically tells you what the parasympathetic division does to things like your eyes and your salivary glands. Whereas on the right, what the sympathetic nervous system does to those same organs. Notice for most things, like the heart and the lungs you see here, there's an arrow pointing for both divisions. So demonstrating once again what we called dual innervation. Most organs are innervated by both divisions. Not everything, for example, the liver, you don't see a green arrow pointing to it. Not everything has dual innervation, but most organs do. And I mentioned that earlier. Okay. So make sure you know this figure. I'm not going to put the figure itself, just the information from it. Okay, and then final slides here will be talking about everything kind of put together for these last several slides and what we call integration and control of autonomic functions. Okay. Thinking back to Tuesday, we talked about reflexes like the stretch reflex, elicited like when you sit on the table and you tap on the patellar tendon and your leg kicks out. Flexor withdrawal reflex, like when the guy stepped on a tack. Those were all described as somatic reflexes. Somatic reflexes involve skeletal muscle because that's what the somatic nervous system controls. Well, autonomic reflexes control your cardiac muscle, your smooth muscle, and your glands. Okay. And just like somatic reflexes, we have a reflex arc. This time we call them an autonomic reflex arc. Okay. And it's basically the same 5 components that we saw the other day: receptors, sensory neurons, integrating center, and the gray matter of either the brain or the spinal cord, and then motor neurons carrying the signal out to effectors. Okay. Well, in the case of autonomic reflexes, these play a lot more complicated roles in the body, something much more complex than like kicking your leg out when you tap the patellar tendon. I'm talking about regulating things like your blood pressure, digestion, urination. Some of these things we'll get into in great detail. For example, we'll spend a couple lectures during the cardiovascular system section of the semester talking about blood pressure, how it changes with like the heart and changes with blood vessels can regulate it. Obviously, we'll talk about digestion and different reflexes and lower that when we get to the digestive system, so on and so forth. Okay. So these are much more complex, but we'll get into all of them later on the semester. I'll run through once again those 5 components of what we'd call now an autonomic reflex arc. First of the 5 are the receptors. Since the receptors just give me the distal end, the dendrites of a sensory neuron, responding to a stimulus, producing a change that will ultimately trigger a nerve impulse. Now, in the case of the stimulus, in this case, it's going to be typically an internal stimulus, a change internally, whereas with the somatic reflexes that we talked about the other day, like stepping on a tack, that's an external stimulus, okay, coming from outside the body, okay. These receptors involved with autonomic reflex arcs are usually classified as interoceptors, and we'll get into those more in our next lecture. When we talk about sensations and sensory receptors, we call them interoceptors because they're located in our interior. So places like inside the body, inside our organs, inside your blood vessels, monitoring, different things in different conditions internally. They become active, which results in an action potential or nerve impulses traveling into the CNS via sensory neurons, where synapsing occurs in the gray matter, which acts as the integrating center. Okay. The integrating center is the processing center made up of interneurons within the CNS that can relay those signals from the sensory neurons to motor neurons. Okay. I would highlight this next sentence. The main integrating centers for most autonomic reflexes are in your hypothalamus and your brainstem. So those very complex things like blood pressure, digestion, urination, those various control conditions, most of the time the integrating, the processing is taking place in your hypothalamus or your brainstem. Okay, which should make sense. We learned the other day your hypothalamus is the major regulator of your autonomic nervous system. Okay, so hopefully that makes sense that we'd send the signals there to process and perform autonomic functions. Some autonomic reflexes, for example, urination and defecation, actually utilizes the spinal cord. So the integration takes place down in the spinal cord, and we don't have to send the signal up to the brain and then back down to help with regulating, peeing and pooping, those 2 processes. Okay. Now, in the case of the somatic reflex arcs that we saw the other day, we only had one neuron that was going to carry the signal all the way out to skeletal muscle. But as we just learned, both Tuesday and today, no matter if it's sympathetic or parasympathetic, to get to the effectors, it takes two autonomic neurons, what we call the preganglionic and the postganglionic. And that's why it says motor neurons, not just one, propagate nerve impulses along a preganglionic and then a postganglionic neuron, which then carries the signal to the effector. And as we've really been hammered in, and I've hammered it in to you guys, hopefully by now you know whenever it's autonomic, we're talking smooth muscle, cardiac muscle glands, which elicit the actual change or response. Typically, those complex autonomic reflexes and changes elicited by your autonomic nervous system are done without our conscious perception. Okay, regulating digestion, making urine, regulating blood pressure. We don't even know those things are happening. We just know that they are happening, and it's thanks to, that subconscious control. Okay. As previously mentioned, the major control and integrating center of your autonomic nervous system is the hypothalamus. And so it does all these things in response to changes, and those changes are detected by sensor receptors and provide sensory input for things like visceral functions, olfaction, which is a fancy word for smell, gestation, which is a fancy word for taste. Also receives input for changes in things like temperature, blood osmolarity, which is just water levels, and then levels of various substances in the blood. So whether that's like calcium, could be oxygen, could be carbon dioxide, could be blood glucose, many other things. Okay. Now, not sensory, but can help change autonomic stuff will be signals coming from the limbic system relating to emotions. Thinking back to our last lecture, we learned that the nickname for your limbic system is the emotional brain, and it regulates emotions like anger and pleasure and fear and contentment. Okay. Well, how the heck and why the heck does your emotions affect autonomic function? Well, think about what happens to your body like when you get really angry. Is that just really an emotional response? Or does it result in physical things to your body? Okay. Does your heart rate go up? Does your blood pressure go up? Do you sometimes cry? Do you sometimes scream? Does your face get red? Do you start sweating? Okay. Those are physical changes to your body. Those are autonomic things as a result of an emotion. So the limbic system can influence visceral things, even though it's emotion that does affect organ functions. Okay? And as a result of any of those input signals, the hypothalamus sends output information to influence autonomic centers in your brainstem and the spinal cord. Not that I'm going to ask you to know this, but posterior and lateral parts of the hypothalamus help control your sympathetic division. Anterior medial parts of the hypothalamus control the parasympathetic. Okay, and with that said, we are now officially done with unit number 2. Feels like it took us forever, but we made it. From the rest of today on, or from right now to the rest of the day, I should say, we're going to jump into our next lecture where we're talking about sensations. So from here on, we're not going to be covering anything that will be on Tuesday's exam, at least not directly. And I say not directly because we're still talking about the nervous system. Okay. It's just now sensations in the afferent nervous system. We're talking about signals going into the brain, into the spinal cord. Okay. Signals for itch and tickle and hot and cold. What we just finished talking about was the opposite of that. Signals going out from the brain, out from the spinal cord to regulate heart rate and blood pressure and to trigger skeletal muscle contraction when you touch something hot, and so on and so on and so forth. So we basically we're going in the complete opposite direction that we just were talking about. Okay. So first thing I want to do, since we're talking sensations, is define what a sensation is. We see another definition, perception, but we've already seen that before, and I've already mentioned that several times. So I'll give you a second to write those things out, and then I'll read through. Okay, definition of sensation. It's the conscious or subconscious awareness of changes in the external or internal environment. And so if we're saying conscious awareness, That means what we call perception. Interpretation of sensations, primarily as a function of the cerebral cortex. So that's what we have for like when we're itchy, when we're hot, when we're cold. When you step outside today and a snowflake hits your arm or your face, you feel that, you're aware of it because of those functional areas in the cerebral cortex. Remember those different areas of the cerebral cortex, like the primary visual area, primary auditory area. And you guys are using both of those right now. Primary auditory area enables you to hear the words that I'm saying, understand what those words are is something else in your cerebral cortex, but hearing those and understanding words is a perception process. So is processing what you see, whether that's on your notes or up there on the screen. Okay. And those are allowing you to be aware of changes. Typically, when we talk sensations, it's of the external environment. But as we know, we don't have conscious awareness of every sensation associated with the body. Like when, the change of a blood vessel stretch occurs, we can't mentally be aware of that. We can't feel when our blood calcium levels are altered. Okay, and that's because we have subconscious awareness of those changes, and we're talking subconscious is typically in the internal environment. Okay. Now, the nature of the sensation and the type of reaction generated vary according to the ultimate destination, nerve impulses that convey the sensory information to the central nervous system. So in other words, if we have perception of things like touch, pain, vision, or hearing, it's because those signals are traveling to the cerebral cortex, okay, to their specific region in the central nervous system. But not all sensations get transmitted to the same place in the central nervous system. So that's why we don't have perception or that conscious awareness of them. So each sensation and where it's processed in the brain is different. Okay. Now, speaking of each unique type of sensation, such as touch, pain, vision, or hearing, those are called sensory modalities. And they can be grouped into two classes. We'll spend the majority of the lecture talking about the special senses. We won't get to that point probably today, but there are 5 special senses: smell, which goes by the term olfaction; taste, which also goes by the term gustation; vision, that's sight, that's pretty common sense; hearing, fancy term for that is audition; equilibrium or balance, okay, is the 5th. The generals are the other 2, or the other class, and there are 2 types of those: somatics and viscerals. Viscerals, called viscerals because they provide us information about the conditions of our internal organs. The word viscera or visceral is just another way to describe your internal organs. So it's telling you the conditions like inside your blood vessels, inside your stomach, inside of the cerebrospinal fluid in your brain. We won't really talk too much about those in this lecture. Okay. What we'll talk more about, and it's not great detail because we'll, like I said, spend more time talking specials, are the somatic senses. Somatic senses include what we call the tactile sensations: touch, pressure, vibration, itch, and tickle; thermal sensations, which would be warm and cold; pain; and then proprioceptive sensations, which allow us to have what we call proprioception, like be aware of where our body is, how it's oriented, without looking at your elbow, you can tell if it's bent or straight. Okay, gymnasts use their proprioception, enabling them to kind of see and feel where their body's at as they're in the air and get ready to land, so on and so forth. Okay. Before we get into any of that, we got some kind of background stuff on sensations to talk about. And the first thing I want to talk about is just how a sensation works, the process involved with a sensation. It all begins. It doesn't matter what the sensation is. It could be cold, like what's shown at the bottom. It could be hot. It could be pain. It could be pressure. It could be sight. It could be hearing. It all begins in what we call a sensor-receptor. A sensor-receptor can be a specialized cell, or it could be just simply the dendrites of a sensory neuron. In this case, these dendrites, which are stimulated by something cold, triggers a generator potential, which then results in action potentials or nerve impulses propagating into the CNS, which this happens to be a unipolar neuron. And I know that because the cell body's off to the side. Okay. So the general purpose of a sensor receptor is to convert the energy of a stimulus, like in this case cold, into the energy of a nerve signal. Okay, so in other words, take a cold stimulus, create action potentials or nerve impulses, so that our central nervous system can detect and interpret the stimuli acting on and within the body, and if necessary, respond. In this case, if we're experiencing cold stimulus, we might respond by shivering. Okay. In the central nervous system, the process of converting stimulus energy to nerve energy is called sensory transduction. You'll see this word transduction or transduce. And as you can see here on the next page, it just means to convert. So converting from one form of energy to another. All right, there are 4 steps involved with the sensation response. So for a sensation to arise, the 4 following events have to typically occur. Okay, this is just an easy situation, easy stimulus of looks like, maybe like the back of a pen pushing in on the finger, and that's going to stimulate receptors down there in the finger. Okay, and so that's number 1, stimulation of sensory receptors. Okay, an appropriate stimulus must occur within the sensory's receptor field, which is the body's region where stimulation activates the receptors and produces a response. Okay, in other words, the receptor field is from this side of the neurons, dendrites, to this side. Okay, in order to stimulate this receptor, these receptors, these dendrites, the stimulus has to be stimulating from here to here, or somewhere in between. If the stimulus was over here, this neuron would not be stimulated. Okay, we'll talk more about receptor fields here in just a little bit. Okay. Number 2, transduction or transduce. The sensory receptor will transduce or convert the energy into stimulus, or excuse me, energy in a stimulus into a graded potential. Remember, a graded or local potential is just a small change in resting membrane potential. A specific strength of the stimulus must be met for the signal to be propagated. Okay, what that sentence right there means is we have to reach threshold in order for an action potential or action potentials to be created. Remember what was threshold? -55. Okay, and remember what happens when you hit -55? Rapid depolarization due to the opening of voltage-gated sodium channels, then rapid repolarization due to the opening of voltage-gated potassium channels. If the stimulus is not strong enough to get us to threshold, can we create action potentials? No. Okay. So that's what we mean here, a specific strength of stimulus. So in other words, we have to reach threshold for the signal to be propagated. Okay. Number 3, I basically just described that. When the graded potential in the sensory neuron reaches its threshold, it triggers one or more nerve impulses or action potentials, as you see they're labeled down there, which then propagate to the CNS. These neurons that propagate the impulse are called first order neurons. So first order neurons would be what you see here. This is an example of one here in blue. Okay. Typically, to get to their ultimate destination in the brain, it takes a few neurons in succession. In this case, this will be the first order neuron. Guess what the name of this purple neuron will be? Second order neurons. Guess what the name of this green neuron will be?
I'm gonna say third.
Third order neuron. Sometimes it takes 4, maybe even 5. Okay, to get to the ultimate destination. And speaking of the ultimate destination, number 4, that's all about, integration of that sensory input. Particular region of the CNS receives and integrates the sensory nerve impulses. If we have perception or conscious awareness of that sensation, it's going to involve the cerebral cortex and a very specific part of it. In this case, it would be the postcentral gyrus. But as I mentioned, if this was a different sensation, maybe it's not touch, but instead it's like something associated with like blood pressure or stretch of your stomach, signal might not be going to the cerebral cortex. It might be going somewhere like to the thalamus or hypothalamus, excuse me. Okay, for the next, 5, 6, 7 slides, we're going to now talk about the different receptors that we find in the body. We use several structural and functional characteristics of these sensory receptors and group them into different classes. There's kind of 3 major ways to classify receptors. The first is the simplest, in my opinion, and it's just based on what they look like under a microscope. Okay, here's what those three look like if you look at them under a microscope. Okay, then we can look at them based on what they detect as far as the stimulus goes, and then basically where they're located in the body with regards to the stimulus that's activating them. We'll get into that here in a little bit. But first, let's talk about how we classify them microscopically. The first two, free nerve endings and encapsulated nerve endings, are basically the same thing with one slight difference. In the case of free nerve endings, it's just the dendrites of that unipolar neuron where they're uncovered. Whereas with an encapsulated nerve ending, the dendrites have a capsule around them. Pretty simple difference there, right? Okay. We find free nerve endings abundant in places like our epithelial and connective tissue. So you'll find them like in your skin or inside like the lining of your mouth. Okay, that includes warm receptors that respond to rising temperatures, cold receptors that respond to falling temperatures, so getting warmer, getting colder. Nociceptors, what we call those, are pain receptors. Easy way to remember, nociceptors are associated with pain. I always ask the question, would you like to stimulate your nociceptors? Most people would answer no, because pain is not typically something that people enjoy. Some people do, but it's not what I'm talking about. Most people don't. And then hair plexuses, which for you guys in anatomy, remember hopefully that those are the ones that wrap around your hair follicles and enable your hair to essentially be an extension of your sensory nervous system. And that's why even though your hair that you can feel on the outside of your body, like the hair on top of your head, even though that's dead tissue, you can still feel that touch because these nerve endings are wrapping around your hairs. And encapsulated nerve endings are nerve fibers wrapped with a capsule. Either it's glial cells. Remember, glial cells would be like those Schwann cells and those satellite cells and astrocytes and whatnot, just supporting cells, or it's connective tissue. Okay, and the function of the encapsulation is to increase the sensitivity, so it's easier to hit threshold. And specificity, we'll talk about what that is. It refers to being able to locate where that sensation is coming from, which I guess I thought that sentence was really important, so I put it on there twice. Very different than those first two are the third type of receptors, which are not neurons, nor are they dendrites of neurons, but instead they are a completely separate cell from neurons. I guess that's why they call them separate cells. Sometimes you'll hear me call them special cells because they are associated with special sensations. Not all of our special senses, but they are associated with hearing and balance or equilibrium. The hair cells in our ears are responsible for that. We have gustatory receptor cells. That's actually what was pictured there on that slide. Those are used for taste. And then in your eyes, in the retina, we have photoreceptors responsible for vision. You guys ever heard of rods and cones before? Rods and cones are examples of separate cells. Okay. Basically, they aren't neurons, but as you can see here, they release neurotransmitters that activate the neurons. Okay. So they have to first become excited, which then triggers a release of neurotransmitters into a cleft that then activate the receptors on this sensory neuron. Okay. No matter what, if you're dealing with free nerve endings, encapsulated nerve endings, or separate cells, the result is still the same. Action potentials or nerve impulses being propagated into the central nervous system. Now, in the case of free nerve endings and encapsulated nerve endings, they utilize what we call generator potentials. Whereas with separate cells, they utilize what we call receptor potentials. Okay, and these are just two types of graded potentials that are created in response to a stimulus. Receptor potentials happen in separate cells. Generator potentials happen in free nerve endings, encapsulated nerve endings, and then in olfactory receptor cells, okay, which olfactory receptor cells are in the nose and are associated with the sense of smell. Okay, back on this slide, I said that separate cells are associated with hearing, equilibrium, taste, and sight or vision. Remember, there's 5 special senses. Smell's not listed here because smell doesn't use separate cells. Cells associated with smell don't create receptor potentials, they create generator potentials. Okay. We call them generator potentials because they directly generate action potentials. We kind of just highlight that last sentence. It's kind of the easiest way to remember it. Basically, you're directly stimulating the neuron, which means that you're directly getting ready to and possibly can generate nerve impulses or action potentials. Whereas with these guys, these separate cells, you have to first stimulate the receptor cell, which then stimulates the neuron. Okay. Once again, the same result occurs, propagation of nerve impulses in the central nervous system, but here it takes a little bit of extra work to get to that point. We have to first stimulate these receptor cells, okay, creating a receptor potential, which triggers the same stuff that we learned about previously. Synaptic vesicles going through exocytosis, releasing neurotransmitters into the synaptic cleft, come across the cleft and bind to receptors on the other side. That's what triggers the action potentials in the first order neuron in this case. Here we're directly stimulating the neurons. Here, same thing. So we're directly generating nerve impulses. Here it's a little bit more involved. Once again, still the end result is the same. Doesn't matter which one you start with, as long as it's being propagated in the central nervous system. Okay, so that's the first way to classify receptors. A second way is based on the type of stimulus that they detect. So in other words, what stimulates them? Most stimuli are in the form of mechanical energy. So it's a mechanical change. These would be loaded with those mechanically gated ion channels that we previously learned about. That includes things like sound waves, pressure changes. We're going to have those like, for example, in our ears for the sound waves. Pressure changes, those will be like the ones in our blood vessels monitoring blood pressure. Electromagnetic energy is another stimulus, like light or heat, or even chemicals. So chemical energy, like in a molecule of glucose. So there are, 6 different categories here for these sensor receptors. The first, mechanoreceptors, called mechanoreceptors because it's mechanical stimulus that activates them. And mechano for mechanical, okay? So that would be like deformation. Deformation just means like the cell gets deformed. A stretching, bending of cells is another way to stimulate these guys. So these are what are going to provide sensations for like touch, pressure, vibration, proprioception. Once again, that's that 3-dimensional awareness and actually hearing and equilibrium as well. Okay. They also monitor the stretching of blood vessels. So in other words, they're monitoring blood pressure, and they monitor like the stretch of your stomach. They monitor the stretch of your bladder. Because like when your stomach gets stretched, your stomach starts to contract its muscle and starts digesting. Okay? Your stomach's empty, it's not stretched, it's not going to be releasing juices, it's not going to be contracting. Okay, but as it fills up, it stretches. These stretched sensitive mechanoreceptors become activated. Chemoreceptors, chemo for chemicals. That's what activates them. Either the amount or the changing concentration of certain chemicals. And this could be internal or external environments. We have these in our mouth, our nose, and various body fluids. Taste is a sensation associated with chemoreceptors. The chemoreceptors in our blood monitor things like carbon dioxide and pH, blood glucose, and smell involves chemoreceptors as well, as we're smelling chemicals, we're tasting chemicals. Thermoreceptors are activated by changes in temperature. The examples there are the warm and cold receptors in our skin. Once again, nociceptors I mentioned earlier are going to be associated with pain, so they respond to painful stimuli. And pain comes from like physical or chemical damages to tissue. Photoreceptors, those are the ones in our eyes that respond to light stimuli. And then osmoreceptors are activated by changes in osmotic pressure of body fluid. So in other words, like water levels is what osmotic pressure really is. And then the final way to classify or group up sensor receptors is based on where the receptors are located and then where the stimulus is activating them. Okay. The first two are basically the opposite of one another, exteroceptors and interoceptors. Exteroceptors are located near the external surface of the body. What is the most external part of your body? Your skin. Okay. Another word for your skin is cutaneous. So any of those receptors in your skin would be exteroceptors, and they are sensitive to stimuli originating outside the body and provide information about our external environment. So they provide sensation signals for things like pressure, touch, pain, and temperature. Guess where interoceptors are? In our interior, okay? And they monitor the internal environment, provide our body with information regarding it. Other term for interoceptors are visceroceptors because they're in our viscera. Places like our blood vessels, visceral organs that would be like your stomach and intestines, bladder. Muscles even have visceroceptors or interoceptors, and your nervous system actually has them as well. And then proprioceptors are those that are located in muscles, tendons, joints, and our inner ear. Provide information about your body's position, the length and tension of your muscles, position and movement of your joints. There's two types, tonic and phasic, and we'll learn about what those terms mean here in a little bit. But they provide positional information on the body or body parts while at rest or even during movement. Okay, so those are the three different ways to classify sensor receptors. First of all, we talked the microscopic structure, how they look under a microscope. What we just finished talking about where they're located and stimulus that activates them, whether that's exterior, interior, or within muscles, tendons, and joints. And then the third, which we looked at second, though, were those that are classified based on the stimulus that they detect. Okay, so that's just kind of getting the nuts and bolts of what types of receptors we have in our body. We have some more background stuff on sensations to talk about before we actually start talking about the different sensations a little bit more in detail. I mentioned several slides ago about what we call receptive field. This is an important aspect of a sensory neuron. Okay, and if I go back to that slide, here is where we were talking about. When we talked about stimulating a sensor receptor, we have to do it within its receptive field. So what is a receptive field? Okay. Well, what constitutes a receptive field varies depending on the type of the sensory neuron involved. It could be as simple as, to stimulate a neuron, you have to be able to stimulate it within the places where its dendrites are. Okay, and that's what we show here is shaded because the dendrites of this neuron span out in this purple shaded area. Okay, we have a visual receptive field, meaning we can only see with each eye a certain amount of space. Okay, if you close one eye, okay, can you see the entire world in a 360-degree lens? No. Okay, we can see from one side to the other side, but not behind us, right? Okay. Well, humans have overlapping receptive fields for vision that enables us to catch things, Okay, if you try to catch something with just one eye open, it becomes very difficult. Okay. Some animals have separate vision receptive fields because their eyes are like on the sides of their heads and on the front of their heads. Okay. And we'll get into more about that. Receptive fields can also be chemicals, like things that we smell or that we taste. Okay. It might have to be a certain amount of chemical to smell it, or a certain amount of chemical to taste it. Okay. Or in like the case of hearing, it could be a set of sound frequencies that cause a response in that neuron. Maybe the sound frequency is something that our ears can't hear. You guys know what dog whistles are? You blow a dog whistle, does that really do anything to humans? No, but it does stuff to dogs, doesn't it? Okay, that's because we can't detect those sound frequencies like a dog can. Their hearing is a lot more depth than ours is, for sure. Now, this is a big paragraph, so I'll read through it, kind of walk you through what's going on, which relates to these figures on the right. In our somatic, sensory, and visual systems, receptive fields are physical areas. The receptive fields of neighboring sensory neurons may be in separate locations, like we show here, or they may overlap. And I was kind of already talking about this when I talked about vision. Okay. If the sensory neurons have separate receptive fields, then a given neuron will respond only if there's a stimulus present in the receptive field associated with that neuron. Okay. So in other words, we've got two sensory neurons here, one in blue, one in green. The receptive field for each of those is circled here. Notice that they are separate from one another. If a stimulus occurs to the skin in this area here, it makes sense that action potentials will be created in that sensory neuron, but not in the one in green. Okay. Makes sense, right? Okay. Think of it like this. If I were playing catch with you guys, sitting here in the room with me, and I threw a ball to Aspen, and she's stuck in her chair, she can't move, just like neurons can't move around. She could only catch the ball if it was, as far from her left to her right arm that she could reach, right? Okay. If I threw the ball closer to Atia, she could catch the ball if it was within her receptive field. Okay. But you both can't catch the ball since you're that far apart. You're in separate receptive fields because your arms aren't going to overlap. If you got closer together, like with Erica, and I threw the ball in the middle of you, chance both of you could catch it, or one or the other could catch it, right? Okay. So if they're separate, we can only create action potentials in the given neuron if the stimulant is present in the receptive field associated with that neuron. Okay. If the sensory neurons have overlapping receptive fields, like we show here, then all participating neurons will respond to stimulus extending into the region of overlap, but it's not going to be equal in number of action potentials. Okay. So looking here where they're overlapping, and we shaded the area that's overlapping, notice the stimulus is occurring here, which is outside of this receptive field. But since there's an overlap between the two, there's action potentials in both neurons, but not the same frequency. There's more more action potentials in this neuron than there are in this neuron. Okay. Does that make sense? So although the stimulus is coming from outside of this neuron's receptive field, action potentials are still being created. Okay. But the response of each neuron is proportional to the relative position of the stimulus. In other words, since this neuron is being more stimulated, more action potentials are being created than there are over here. And we have a high frequency of action potential shown here in purple, very low frequency in here in the pink one. Okay. This is going to help with stimulus localization. Okay. In other words, it helps you locate where a stimulus is acting on in the body. Okay. So like if I walk over and I pinch you on the arm, or I poke you with my pencil, okay, by being able to locate where that stimulus is at, you're going to feel exactly where that pain is. You're not going to say, ow, my arm hurts, and don't know where at in the arm. You know exactly where it hurts. That's what stimulus localization is. Okay. By contrast, when a sensory neuron has a separate receptive field, the brain has no way of knowing exactly where in that receptive field the stimulus is located, and there are no signals from other sensory neurons that can be used for the comparison. Okay. In other words, like if, I was poking you in the skin, like right here where there's this singular neuron, okay, the brain might not know if the pin was poking here or here or here or here or here. Whereas over here, we're going to be able to very precisely know. So think localization like being able to locate. Okay, locate where that sensation is coming from. Okay. Receptive fields frequently overlap in somatic and visual systems, enhancing our brain's ability to localize somatic, sensory, and visual stimuli. So in other words, we can see exactly where something's at. We can feel where exactly something's touching us, where exactly we're being tickled, where we're, stepping on that Lego, where that pain's coming from. Okay. So localization, and we'll talk a little bit more about that here in a minute, is very important, involved with receptive fields. As you've already learned, sensory information is ultimately converted into action potentials or nerve impulses, whatever you want to call them, before they're eventually conveyed into the CNS for integration or processing. If all action potentials are the same, how does the nervous system tell the difference between one stimulus and another? For example, how does our brain know that we're smelling something and not tasting something? How does our brain know that we're itchy instead of tickled? How does it know when we're hot instead of cold? Okay. This is accomplished by what we call sensory coding, using organizational and functional features of the nervous system to represent specific details about a stimulus. And encoding or sensory coding involves 4 attributes of a stimulus: the modality, the location of the stimulus, the intensity of that stimulus, and the duration that it's acting on the body. So let's first talk about the modality, which we learned earlier today. Modality just refers to each different type of sensation. Touch, pain, vision, taste, hearing, those are all different modalities. And we encode each of those different modalities with a different, what we call labeled line. And we process those in different parts of our brain. Okay. Looking at the figure on the right, we've got all sorts of different sensations illustrated. We've got in the mouth, gustatory receptor cell, that's going to be carrying signals for the sense of taste. Touch receptors in our hand or in the skin, doesn't matter what part of the body, I should say, responding to the sensation of touch. Photoreceptors involved with vision, hair cells in the ear for hearing, olfactory receptor cells in the nose for smell. Okay. Those different sensory signals are being conveyed to different parts of our cerebral cortex for perception. Okay. Remember that figure on our central nervous system lecture where all those different functional areas were mapped out? Okay. That's what I'm talking about here. Okay. What we see gets processed in the back of our brain. What we hear is processed somewhere completely else. What we feel as far as like touch goes, processed somewhere else than where we taste it versus what we smell. Okay. And each of these sensory signals is going to involve a different set of neurons that carries that signal to the cerebral cortex. So the neural pathways that convey information about modality from peripheral receptors, specific regions of the cerebral cortex, are called labeled lines. Each of these groups of neurons carrying the signal to the brain is a different labeled line. Okay, it's a set of neurons transmitting information for only one modality. So this labeled line doesn't carry the sensation of touch. It only carries the sensation of taste. This one only carries the sense of touch. This one only carries the sense of vision. Okay, and they go from where they originate, and that's why you seem to see with your eyes, hear with your ears, or feel pressure on your arm, despite the fact that you're processing those sensations up in your brain. You're feeling them where that sensation is coming from, right? Because the sensation is originated in receptors in those different parts of our body. Okay. And because stimulation of receptors in each of these parts of the body activates a separate labeled line, we then carry that signal to a specific region of the cerebral cortex that then perceives a modality associated with that labeled line. The association of a modality with the activation of a particular labeled line is called labeled line coding. So that's how we can tell sensations apart. This is why we can smell and just determine that's different than taste. Although those two kind of go together. We can feel itchy and not tickled. We can feel hot and not cold, and so on and so forth. The second attribute that we use to perform sensory coding is the location. Okay, and that involves what we call acuity. This is the sharpness of perception. In other words, it's our body's ability to precisely locate and distinguish one stimulus from another. Okay, we were already kind of talking about this just a second ago with regards to receptive fields, if they overlap or if they don't, like we show here where they don't versus where they do. Okay. In our somatic, sensory, and visual systems, two major factors affect acuity. So in other words, these two things affect how well we can locate a stimulus acting on the body. The size of the receptive field, so how big the receptive field is, and then something that's brand new, which is lateral inhibition. Okay. We'll start up here in the top right, which relates to receptive field size and that overlap. Okay. All right, what they're doing here in this is a pretty cool little experiment. I wish we could do it. If we were all together, we would. It's called a 2-point discrimination test. Okay. Basically, you have these calipers that have 2 points on the end of them. Okay. And these 2 points can be changed as far as how far apart they are. You can put them really close together and so close that they're almost like touching. And then you can spread them apart where they're really far apart. Okay. What you can do is go through and have the person that you're testing this on close their eyes, and you can touch them on places throughout their body. And what you're trying to do is test to see if they can feel both points or if they're still feeling 1 point.
This is how they test you for pain pop.
Probably somewhere around that. Okay. If, for example, we show here where they're testing in the back, both points are within this singular receptive field. At this point, this person will only be able to feel 1 point touching their body, despite the fact that there's 2 little pieces of that caliper touching them. Okay. Whereas over here, doing it on their fingertip, look what's happening. The receptive fields are separate, and those 2 points are in 2 different receptive fields. So this person would feel the 2 points individually. Okay. This graph is showing you it kind of looks backwards. The smaller the bar here, the lower the line is, the more sensitive that area of the body is. Okay. The bigger the lines mean that you have to spread that caliper really far apart to actually feel those 2 points. Okay. And it all has to do with the size of the receptive field and if they overlap one another. Okay. So let me read through and hopefully it'll make some sense. The size of the receptive field, so how big the receptive field is, varies inversely with the number of sensory receptors that it contains. So if something is inversely related, it means if one goes up, the other one goes down. Okay. So in other words, the bigger the receptive field, okay, the fewer the number of receptors in that area. Okay. You can all hopefully agree with me that the receptive field of these 3 is a lot bigger than the receptive field of each of these 5 over on the right. Okay. Very small receptive fields, but these receptive fields are very closely together, and there's a lot more neurons here than there were here. Okay. And since there's so many here, there's going to be more overlapping that exists, whereas here they're all separate. Okay. So the smaller the receptive field, the more densely packed it is with sensory receptors, and the greater its acuity, the greater the ability to precisely locate and distinguish one stimulus from another. By having the tip in this receptive field and this receptive field, then that person's going to easily detect, okay, there's 2 points, not just one touching my skin. Okay. Whereas with the larger receptive field, you have to really separate these calipers apart to where 1 point is here and 1 point is here, and 1 point's here and here, to actually feel the 2 different points. Now, why is this important? Well, look at the ones that have the small measurement associated with them. The fingertip of your index finger, the lips, your big toe, your palms, your forehead. We can describe those places as the most sensitive, especially your fingertips and your lips. What do you do with your fingertips and your lips? You touch and you feel things with them, right? So it makes sense that you want those places to be the most sensitive, to be able to interpret sensations the most. Okay. When you go to feel something, are you going to rub the back of your calf on it? Rub the back of your arm
No, I
on
did
it?
my cheek.
Okay. You might do your cheek. Your cheeks are very sensitive. Okay. So the more sensitive places are going to be the places that are feeling and touching things. We touch stuff to our lips before we put it in our mouth. Okay. When you kiss a baby, okay, that sensation is very pronounced because your lips are super sensitive. Okay. Does that make sense? Okay, in lateral inhibition, and that's what we show down here. Input from sensor receptors along the border of a stimulus is substantially inhibited compared to input from sensor receptors at the center of the stimulus. Okay, so it looks like a pencil's pushing in on the skin, not so much that the person's getting stabbed, but they're feeling a little bit of pressure. Okay, you guys can all hopefully agree with me that the purple neuron here at the center is being most stimulated, whereas although the green and the blue neurons are being stimulated, we're more directly stimulating the purple one, right? Okay, and that's definitely what happens. A lot of action potential are created in the purple neuron, a few in the green, and a few in the purple. Okay, but look what happens as the signal gets propagated inward from that first order to the second order neurons. Okay, due to lateral inhibition, the neurons on the side that are stimulated but not nearly as much create inhibitory signals, and those are shown down here with these inhibitory interneurons that inhibit their second order neurons that don't really affect the second order neuron of the straight on purple neuron. Okay, so less signals end up getting propagated via these two here versus here we can see more action potentials. Okay, so less action potentials means these are being less stimulated than this one, and therefore our brain's going to know, okay, this is really where that point is touching the body. Make sense? Okay. All right, let's talk stimulus intensity. All right, before you do that, I'm gonna have you stick a pencil in your non-dominant hand and just hold on to it. Just a pen or a pencil that you're not using and just hold on to it. Okay. How can your sensory systems detect stimuli at differing intensity if all nerve impulses are the same size? In other words, if an action potential for a really firm touch tops out at positive 30 or 35, as it does for a light touch, how can our body feel the difference between light touch and one that's firmer? Okay. Well, the main answer to these questions, and I've already kind of talked about this, is frequency of action potentials. How often or how close together and how many action potentials we create can vary from neuron to neuron. Okay. So frequency, it refers to how often they're generated at the trigger zone of the sensory neurons. With a light touch, which results in a lighter stimulus, as far as strength goes, that's what we're measuring down here, light versus more pronounced strength stimulus. Okay. We create less action potentials, and the action potentials are spread farther apart, versus with a higher intensity strength, more frequently are we creating action potentials, and this would be like what happens with a firmer pressure. So light touch means a lower frequency and therefore less action potentials. Firmer pressure would result in more frequent action potentials and therefore a more higher number of action potentials. In addition to this frequency code, a second factor is the number of sensory neurons that become activated or recruited by that stimulus. Like in this case here, This would be like just lightly pushing in on the skin, affecting just this purple shaded area of the skin, which means that really only this neuron becomes activated. Whereas if you really push in on the skin, now that whole area of the skin is activated where all three neurons become activated. And so that's how you can detect, okay, yes, this feels like really strong pressure pushed up against the skin. This feels really light as far as touch goes. Does that make sense? Same thing could be for like smell or taste. Okay, there's a whole lot of chemical. You're going to recruit more neurons. It's going to smell a lot stronger. If it's a really, really weak amount of chemicals, you're probably just going to barely smell it. Now, speaking of smell, smell is something that our body undergoes adaptation with. And that relates to the duration of the stimulus. This will be probably our last slide. Okay, stimulus duration is encoded by the duration of action potentials in the sensory neuron. In general, the longer the stimulus lasts, the longer the sensory neuron produces action potentials. Okay, so that makes sense. If something's acting on the body, our body responds by creating action potentials as long as that structures or that stimulus is acting on the body. Okay, if something's touching us, we're going to feel that touch for as long as we're touching us, right? That would make sense. Is that actually what happens? No, it's not always what happens. Okay. If a stimulus persists, however, most sensory neurons do not continue to generate the same frequency of action potentials because their sensory receptors adapt. Adaptation is a process in which the generator potential or receptor potential decreases in amplitude despite being in a maintained constant stimulus. Okay. This is what can cause a sensation to disappear or to fade or disappear, even though the stimulus persists. Okay, just a second ago, I told you to hold on to your pencil. Had I remembered that, I told you to do it 10 minutes ago, or sometimes I have students stick it behind their ear. Okay, and as you know, we go on teaching, you might forget that that pencil is in your hand or in the back of your ear. Okay, I assume everybody got dressed this morning because you're all wearing clothes, right? And when you put those clothes on in the morning, you feel those clothes on your skin. But do you feel that touch of that clothing to your skin all day long? No. Okay. But that's a constant stimulus. That skin is always being touched by your shirt or your watch.
I forget my watch.
How many people have ever forgotten where their cell phone is despite the fact that it's in your hand? Or lost your sunglasses when they're on top of your head?
Yeah.
That's because your body's going through adaptation. Okay. So here's two examples of adaptation at the bottom. Okay, we're looking at two different graphs. Here on the left is a slowly adapting receptor. Here on the right is rapidly adapting. Okay, it's kind of a three-part figure for each. So I'll walk you through. Okay, this blue segment here is showing you when this sensation begins, because since the stimulus becomes active here, it's turned on here, and then it's turned off here. Same thing here and here. Okay, in a slowly adapting receptor, as soon as that stimulus acts on the body, our body starts creating receptor potentials. Notice that they start to decline. But during that on until that off point comes, we're creating action potentials continuously. Although as the receptor potential decreases, the frequency decreases. Okay. Whereas over here, we hit that on, we create an on response, action potential. But during this middle section, that receptor is still being stimulated, but the receptor is not responding by creating action potentials until the off response is triggered. So this type of receptor experiences adaptation. Okay. They generate a burst of action potentials when first stimulated, but then quickly adapt. The sensation is going to disappear. Okay. It'll sharply reduce the signal transmission, even if that stimulus continues. Okay. Sensations that are going to involve these types of receptors would be like for pressure, touch, smell. Okay. For those of us who are in Atchison, there are days here in the wonderful city of Atchison, where you walk outside and it hits you like a truck, how stinky it is here. Okay. But if you stand outside for 5-10 minutes, does it continue to stink?
No.
It does, but we don't smell it anymore. Okay. As kids, you go to the swimming pool, as soon as the summer hits, the water's like 70 degrees. You jump in the water and you're cold. Are you cold the whole time, or do you get used to it? You get used to it. Okay. Has it heated up to the point where your body's not cold anymore? No. You're still experiencing a change to the body. Your body's just not continually sending that signal into the brain to process that. Okay. For slowly adapting or tonic receptors, they adapt much more slowly. So generating action potential still continues, and it will do so as long as the stimulus persists. Although as it gets longer and longer into it, frequency of that action potential number goes down. Things that slowly adapt as far as sensations go would be like pain, body position, chemical composition of the blood. Think about those. How awful would it be if, for example, our blood sugar dropped tremendously and our body detected that signal right away, sent a signal into the brain, but we didn't fix it? Our body just decided, okay, I'm not going to send any more signals to your brain to tell you our blood sugar is low. Or if you've got, a nail sticking in your leg and you feel that pain, but then 2 seconds later, you don't feel that anymore. So things that don't rapidly adapt are very, very important signals. We have to continuously send those into the brain because they deal with very, very drastic consequences if our body doesn't respond. If you don't continually send signals in for your body position, you're
So having people that don't feel pain, don't feel pain.
a lot of other things, a lot of factors go into that. Could be nerve damage. A lot of times it's some type of nerve damage and that kind of stuff. All right, we're going to stop here. This is going to be a nice spot to stop. We'll pick up again with this on Tuesday after our exam and go from there.